Good morning, afternoon, and evening. It's Tuesday, April 7th, 2020, and on behalf of everyone at Simulations Plus, welcome to today's webinar. As the globe continues to grapple with increasing challenges to sustaining business as usual, we hope being able to provide our scientific talks and scheduled poster presentations for the now canceled SOT Tox Expo and Annual Society of Clinical Pharmacology and Therapeutics in a digital format is a valuable experience for you. Presenting today will be Brett Howell, Vinal Lakhani, Nadir Hamzavi, with a DSX demo at the end by Corey Berry. It's going to be a very busy 90 minutes. An opportunity to ask questions will follow Brett's talk and after each poster session. You may send your written question using the questions pane on your control panel, or you may use the hand-raising icon on your control panel to ask questions directly. Please be sure to enter in your unique audio PIN displayed when you join the call. This call is being recorded for playback on www.simulations-plus.com. Now, before we get started, we have a quick poll question for you. Do you use modeling and simulation in your current research activities? We'll give you 30 seconds to answer the question. We're getting a strong response to yes, so you guys are in the right place. And about 10 more seconds left. Great, we're going to close the poll now. And it looks like 85% of you have said yes, you are currently using modeling and simulation in your current research activities. Now it is my pleasure to introduce Brett Howell. Brett is the president of DILIsym Services, developing and using DILIsym to improve the development of safer and more effective drug therapies. He's the associate director of the DILIsym initiative since its inception in 2011.
Thank you, Arlene, and thank you everyone for joining us today. I'm excited to share this presentation of modeling drug-induced liver injury drug-drug interactions, which I'll be referring to as DILI drug-drug interactions with DILIsym, our computational quantitative systems toxicology platform. This presentation was originally planned for the Society of Toxicology SOT Lunch and Learn. So if you are interested in receiving your free lunch, since we weren't able to provide that to you, please contact Dan O'Connor or John DeBella. I'm sure they'd be happy to provide you with a free lunch. Next, please. So as we get started, I'd like to take just a moment to give everyone a brief introduction to our offerings at Simulations Plus. Across our family of companies, Simulations Plus, Cognigen, and DILIsym Services, we have a wide array of products that we both license and also use for consulting. Our products and our offerings span all the way from discovery to clinical, as you can see on the slide. We do quite a bit of machine learning and in silico molecule optimization, absorption, and PBPK modeling, quantitative systems toxicology and pharmacology, which will be the focus of my presentation today, but also clinical pharmacometrics modeling, PK/PD modeling, using Kiwi, our non-MEM front-end interface. And you may have seen in the news that just last week, we were excited to acquire Lixoft. So we now offer the Lixoft Monolix suite of software tools as well. So we hope that you'll reach out to us regarding one of our many offerings. Next, please. So one of the things that's fantastic about Simulations Plus is that our tools fit together strategically. And today I'll be showing you how that happens with ADMET Predictor feeding into GastroPlus, feeding into DILIsym and RENAsym. These are two of our quantitative systems toxicology tools that help predict whether or not a drug will be harmful to the body. And ADMET Predictor and GastroPlus allow for the prediction of drug concentrations, which enable the use of these tools. So you can see how these tools strategically fit together. Next, please. So now, DILIsym services, while quantitative systems toxicology was really our founding and initial interest and what I'll be focusing on today, we also do a lot of other things, and primarily quantitative systems pharmacology as well. And so I would be remiss if I didn't mention that we work in the areas of idiopathic pulmonary fibrosis, NAFLD and NASH drug development, and also radiation, and most recently, not listed here, is heart failure. So we'd love for you to get in touch with us. We license all of these platforms out so that you can use these platforms within your own groups to help make decisions and inform your drug development process, or we will work with you on a consultation basis. Next, please. First, let's answer the question here on slide 5 of what is quantitative systems toxicology. It starts with a mathematical representation of the relevant biochemistry or pathophysiology. In this case, you can see small depictions here of both DILIsym and RENAsym. So these are mathematical constructs that reproduce drug-induced liver injury pathways that occur in the liver or drug-induced kidney injury pathways that occur in the kidney. If you click it again, we then combine that mathematical construct with the ability to predict the drug exposure, both at the site of action and systemically. And lastly, we use, if you click one more time, we use in vitro assays or potentially machine learning models to predict the potency for each molecule, both parents and metabolites, to affect a given tox pathway or mechanism. Next. So once you put all of that together, we're able to actually predict outcomes in a human and determine whether or not the drug will be safe or unsafe for that particular organ or system. Next, please. Now today, the title of the presentation, and of course the focus, is DILIsym, our drug-induced liver injury product. But I did want to take a moment to first address, next, the question of not only what is quantitative systems toxicology, but how can it actually help your organization? First of all, these tools can help keep patients and also volunteers safer, which is of course the most important thing. Second of all, these tools can be used to predict liabilities before they happen, which can save your company money and also eventually save patients money. Next, they can help you choose the lead candidate that's most likely to succeed from a safety standpoint. And lastly, we often use these tools to help communicate with regulators. A lot of companies are requested to do due diligence to investigate findings, and these tools are great tools for doing that in a very organized and efficient manner. Next, please. Now today I'll talk about DILIsym, and on Thursday you'll hear about GastroPlus. And I just wanted to mention here briefly that in the future, hopefully next summer, we will be offering DILIsym and RENAsym, both of these products, from within GastroPlus as integrated products. So I hope that you'll stay tuned for that development. We think that this will make the workflow and the use of all of these tools collectively much more efficient, and we're really excited about the release of the integrated versions. Next, please. Now, I want to start by introducing you all to RENAsym very briefly and mentioning how it can be used and the origin of the product, and then we'll get to DILIsym. RENAsym is a product that helps predict acute drug-induced kidney injury. We're working towards the development of this product now, and we will be releasing it next summer. Next slide, please. As a company with a history in drug-induced liver injury, obviously, we knew that we were well suited to turn our attention to other organs or systems, and the kidney was a natural fit for us. In fact, we had many customers requesting that we move to focus on the kidney. Next. And of course, our experience in managing quantitative systems toxicology frameworks, the development of those software platforms and consortia related to those platforms was important for our movement into this area. So we decided to move into the kidney area with RENAsym, to click it twice. And to that end, we received a large NIH grant to help fund this effort. So the good news is the product development is already underway, and that's being funded through a government grant. Next. Now, to get started on this process, we first went out and hired key personnel. So Yeshi, Gebremichael, the first author on this kidney injury publication, came aboard and joined DILIsym services. And we also reached out to Melissa Howell, the last author here, and been working with her on a consulting basis. Next, please. To give you an idea of the scientific content behind the first version of RENAsym, you can see here on the slide, if you focus on the diagram on the bottom right, a series of mechanisms of kidney injury that come together within the software. So some of the key ones that I'll highlight include reactive oxygen species, also known as oxidative stress, direct effects on mitochondrial function, mitochondrial DNA damage, and also crystal nephropathy. These are some of our initial key mechanisms that we're including, along with accompanying biomarkers and an immune response. We're also representing a number of exemplar compounds to help us validate that the software connections are correct and accurate. Next, please. The workflow for RENAsym is actually very similar to DILIsym. We start, if you follow here from the top left, with the exposure information. For this, we use GastroPlus to predict the concentrations both at the site of action, so in this case the kidney, but also circulating concentrations. Next, on the bottom left, we use in vitro technology to assess the potency for the molecules on the toxicity pathways. In addition, we can use machine learning models, and I'll talk more about that later. And lastly, on the bottom right, we use information around the types of patients and the dosing protocols that we want to input into the software. And all of that leads us into a simulated outcome of whether or not the drug will be safe for the kidney. Next, please. So here's an early look at some of our results for RENAsym. On the top right, you see the number of viable proximal tubule cells for acetaminophen, cisplatin, and gentamicin. And what we can see is that acetaminophen, the safe compound for the kidney at therapeutic doses, we see no cell loss, whereas for cisplatin and gentamicin, we see a drop in the number of viable cells. And then down below, correspondingly, we see an increase in urinary alpha-GST with cisplatin and gentamicin, but no increase for acetaminophen. So this was an early result from our model. This model is certainly not final yet. We're working over the next year to finalize the product, but it does show that we're able to differentiate between toxic and non-toxic compounds in terms of kidney toxicity. We think this software will be very useful for your programs as well. Next, please. Because we have developed the DILIsym software, as I'll discuss in a moment, through the DILIsym initiative for 10 years now and done so very successfully and enjoyed the collaboration that has brought with industry scientists, we decided to also start the RENAsym Consortium. Now, the RENAsym Consortium allows for the development of RENAsym over time through the collaboration with our company, with all of you. In addition, it's a way to access the software and other exclusive member benefits that straight software licenses don't allow. And I'm happy to announce today that we've decided to include RENAsym Consortium membership with DILIsym Initiative Stage 4 membership renewals as well, or new members for Stage 4 as well. So as I move to the presentation and talk about DILIsym, if you've been interested in DILIsym and now RENAsym piques your interest, this is the perfect time to join because you're going to get membership and access to both products, both consortia, both scientific networks at the same time. Next, please. We also have an excellent scientific advisory board for RENAsym, highlighted by our recent addition, Dr. Frank Sester, who used to be at Merck and also at the FDA, but also many other great scientists and physicians who are helping to direct the effort. Next, please. So without further ado, I want to talk about DILIsym. This is, again, a product to focus on drug-induced liver injury. We released the 8th version of DILIsym, 8A, this past year. We do plan on releasing DILIsym X, which is our next version, our latest and greatest version this summer. Next, please. As I mentioned, for about 10 years, the DILIsym software product has been developed through the DILIsym initiative. And you can see some of the current members in the middle of the slide here at the company logos who are members of the consortium currently. We've had over 19 companies involved in the consortium from the inception and over $10 million invested in the project from those companies. What's come out of this has been a slew of regulatory submissions with the product, which I think everyone that was really the ultimate goal when we all started, was to be able to use this software for regulatory submissions and interactions. We've also published well over 30 publications, maybe up to 35 publications now, focused on methods and applications. And we have a great scientific advisory board who's helped guide the effort, again, recently bolstered by the addition of Frank Sester, but with also 6 other members. And I should say from the inception of the consortium, it's been led and directed by Dr. Paul Watkins, a world-renowned physician and hepatologist. Lastly, I'll mention that the FDA has been involved initially with SAB membership and also direct financial support, and now more recently through actual evaluators and users of the product through licensing the software. So this consortium has been very successful in bringing all these parties together and being a forum for discussion and scientific advancement. Next, please. Now here I've just listed a few of our recent publications. 3 of these, the ones on the right and the bottom, are actually co-publications with our sponsors. So Palladio Biosciences, Pfizer, and also Simpra. And so feel free to check out those publications. They show some of our most recent applications, and Paul also has a great review of DILIsym and clinical applications in liver injuries. Next, please. I won't belabor the points I made earlier about membership in our consortia, but I do want to briefly mention that if you join the DILIsym initiative, membership includes not only software access, and as I mentioned before, also RENAsym Consortium membership as well, a new benefit, which we think is a fantastic deal, but also exclusive access to things like special licenses that typical license holders have to pay extra for. Our higher performance grid license is a feature that we have that enables you to deploy these tools on both cloud or local clusters. And this is a grid license that you get included with membership that you have to pay extra for if you license the product separately as without membership. In addition, we offer members additional training hours and a substantial discount on consulting. So really the package deal of joining as a member is a much better deal than licensing the product or engaging on consulting services separately, although of course those remain options for both DILIsym and RENAsym. But we'd love to have a number of companies join us and join the current members. We are in a renewal stage right now where we are renewing memberships and adding new members for stage 4, which is the 2021 to 2024 timeframe. Next, please. So to talk a little bit about the details of DILIsym and what's under the hood, again, I want to focus your attention to the diagram on the bottom right. The 3 main mechanisms of drug-induced liver injury that we have focused on most over the past 10 years include reactive oxygen species or oxidative stress, also known as ROS, mitochondrial dysfunction and toxicity, and intracellular bile acid transport and disruption. Those have been the workhorses, if you will, for many of the exemplar compounds that we've now simulated and predicted outcomes for. Over 70, I would say, including some of our more recent proprietary and internal projects, 80 or 90 projects where we've simulated outcomes for. But we have begun working in some new areas, and one of those is shown to the left of the diagram here, where you see the cholangiocyte life cycle being affected by the phospholipid to bile acid ratio. So we are that particular submodel is in development and currently being validated. So we do continue to advance the DILIsym software. One of the things that's really nice about this type of mathematical framework is that you can do things that are difficult to do in the laboratory or in the clinic prospectively. And one of those things is to combine different therapies and to determine what happens if you put a combination of drugs onto a framework like this and ask what-if questions that really you would only find the answers to clinically in the real world. It's very difficult to run controlled studies. Next, please. Now, I won't spend a lot of time on this slide talking about DILIsym X because, as Arlene mentioned, there's going to be a great demo here at the end of the presentation today. But I just want to say that we're very excited about the release of the new platform. In addition to the software environment being completely overhauled, which Corey will tell you about, we do have 4 new exemplar compounds going into the software for your use as examples, and we also have a couple of 2 new smaller populations or sim cohorts for your use as well. So we think it's going to be a great release. Next, please. Now, the workflow for DILIsym is very similar to RENAsym. It starts with mechanistic DILI information, which we gather from in vitro systems, typically working with Cyprotex and Solvo for those data collection activities. Now, in the future, hopefully next summer, when we release the integrated GastroPlus, DILIsym, RENAsym products, we will also be releasing accompanying machine learning models. Those machine learning models, the goal is to have those machine learning models predict the potency of the molecules on these key pathways so that instead of running in vitro studies, you can use those models. Now, they'll obviously be less accurate than running the actual in vitro studies, but they'll be able to handle and be much more efficient for large batches of compounds. So the idea is you would use machine learning models to kind of replace this gray box here or figure out the information from this gray box early on in drug development, perhaps for hundreds or thousands of compounds. And then as you move to lead optimization or first in human, you begin to collect these data and improve the accuracy of the input data for the simulation. Next, please. So after gathering some information on the potency of the molecules, on the pathways, we then need to predict a disposition. Again, we use GastroPlus and PBPK modeling within GastroPlus for that activity. And then next, we figure out what dosing protocol we want to use. Perhaps we want to switch the patient types to match the patient types in the clinical study, or we're going to use healthy volunteers. And once we put all of that together, next, we will see a prediction of risk. But it's not just a prediction of risk. It's also an understanding of the mechanisms that might be involved. It's an understanding of the patients that might be at risk. It's an understanding of how you can mitigate that risk. So there are so many things that you can do with these in silico tools, and I think that's really what makes them so valuable and useful. Next, please. So getting into the specifics a little more and showing you an example of one of those so many things you can do, today we're going to talk about polypharmacy or DILI-DDIs. And the concept at first seems sort of simple. So if you click next, you'll see that we have an example illustration here of a drug A hitting the ROS pathway within the liver. And if you click again, we see that, okay, perhaps drug B also hits the ROS pathway. Now, this interaction in and of itself can be complex because depending on where the ROS to effect or outcome response is on the exposure response, you can get a nonlinear or linear response. So these two drugs may actually show you an additive type response where a patient has a little bit of injury and a little bit of injury and add them together and you get what you would expect. Or there are cases where A plus B can equal 20 times C, a synergistic response, in other words. And if you click it again, there's also other scenarios where, for example, a drug C could hit the bile acid transporter pathway, but if you click again, a metabolite for a different drug could actually hit the mitochondria directly. And now you can see the complexities associated with all these different molecules and metabolites coming together, hitting different pathways, and then very importantly, on the back end within the liver, those pathways are actually connected mechanistically in the model. And so this is a very powerful tool for asking what-if questions, especially what-if questions that relate to what could happen when a drug goes onto the market. And all of the patients, we all know that patients are taking multiple types of drugs. So if you click again, so we can look at the effects that can be similar or different when we compare normal healthy volunteers to patients. So that's always a big question with clients is, well, what if I go to a patient group? Do I really expect to see the same thing that I saw in my small controlled healthy volunteer studies? We can also look at the effect of parents and metabolites and how those will mix and match together. Next, please. So today I'm going to focus on a couple of quick examples, and the mitochondrial dysfunction is sort of a linchpin or a focal point for these examples, so I just wanted to highlight that. Next, please. Some scenarios where we have seen DILIsym use be interesting and important for DILI-DDIs include increased ALT elevation frequencies were observed in patients taking 2 drugs, data collection and simulations for each compound in isolation and combined. So this was a scenario where we worked with the model. The second scenario was ALT elevations were observed during a PK-related DDI study, and I'll talk about this today. Thirdly, ALT elevations were seen in patients taking acetaminophen. And lastly, a drug was targeted for a patient population taking an existing drug that also causes ALT elevations. So preemptive simulations are now underway to help answer the question, does that pose a problem for the sponsor? Next, please. The first brief example I'll cover involves valproate. Now, valproate does lead to liver enzyme elevations in approximately 5 to 10% of patients. And if you click again, we'll show that there are a number of in vitro and in vivo data sets that suggest, number one, that mitochondrial effects might be causing this, and number two, that perhaps the liver is able to adapt to valproate because of mitochondrial biogenesis or mitogenesis. And so we included valproate in the DILIsym software. We also included the adaptation mechanism. And if you click to the next slide, you'll see an example of that. So here on the left, we have a simulation of 16 individuals where we've dosed them with valproate and we've titrated them up to a dose that is similar to what's done in the clinic. The titration is also similar to how it's done in the clinic. And what you see is a fascinating response. Again, this is a simulated response. This is not clinical data. You see all of the individuals, or most of them, with a bump in their ALT or their liver enzymes. It's a small bump. And then they resolve and go back to normal. And that's with continued treatment. And that's very much consistent with what we see clinically, qualitatively. If you click it again, you'll notice this is a new drug, Compound X. This is a drug that we're not able to show here, but this is a real simulation of a real compound. And what we see is that when we give Compound X, we get 4 of the 16 individuals showing a small bump in ALT, not much ALT. And so the significance of the ALT is relatively minor, but you do see some movement in some of those individuals. But if you click it again, now you see something interesting. If you give those patients valproate and you allow them to reach a new steady state, and then you give them Compound X, now we see all 16 of those individuals showing a small bump in ALT. And if you click one more time, this actually represents a confidential case in which we're currently now working where 2 compounds, one being valproate and then a second compound put on top of that, leads to a synergistic response. And what we're able to do with the simulations is help the client understand why that response is occurring. Next, please. The second example actually relates to a very common compound, metformin. So a client was running a DILI-DDI study, and if you see the diagram here, there's a denotation for day 1. So the client started dosing their compound Z, on day 1, and then on day 14, they added a single dose of metformin. Metformin is a very safe drug. This is a typical DDI protocol, or at least not too uncommon DDI protocol. But if you notice the little lightning bolt in the picture there, they saw liver enzymes a few days after dosing metformin. So the client came to us and said, do you really, can you use your simulation tool to help us understand whether or not it's even conceivable that maybe metformin actually somehow led to this response? So if you click it again, you'll see 3 simulations which led us to the conclusion that yes, metformin could be a part of this response. First of all, for their compound Z only, we see very minor ALT elevations, 1 minor increase in ALT, that's the top left. But then on the top right, we show a simulated metformin only prediction, and we see no movement in liver enzymes. So there's no injury to the liver. Then on the bottom left, now we give the compound Z for 14 days, and then we add metformin, as was done in the clinic, and we see a synergistic and a very clear response in at least 2 of the individuals. And it turns out that the effect was due to an interaction between mitochondrial dysfunction and bile acid transporters. Next, please. Now, hearkening back to the prior example, we did have discussions with the client about why in the clinic they were seeing significant ALT elevations, but say clinically benign cases where patients were okay. They were not having severe symptoms. Whereas in our simulations on the top here, you can see the FDA's preferred way of looking at liver safety data. The EDISH plot, which is bilirubin versus ALT, you can see that there's an individual in each of our SymPops in the top right quadrant. And to summarize briefly, you don't want to be in that quadrant. It's the unsafe quadrant, the Hyde's Law quadrant. So we added mitochondrial biogenesis or adaptation to the simulations, and then you can see the result here on the bottom right-hand side, where in all 3 SymPops, the patients are now adapting, so they do have significant liver injury signals, but they do not progress on to significant liver injury because of the adaptation. And this very much matched with what they saw clinically. So I think what these two examples hopefully gave you a flavor for is how you can investigate and even prospectively, preemptively simulate what your compound might do with other compounds, either on the market or in clinical studies where they're both going to be dosed, and help determine, are there interactions between those compounds that need to be taken into consideration. And sometimes it's a matter of avoiding those other compounds, sometimes it's a matter of just adjusting the dose, and sometimes it's just a matter of being aware so that you know where to look in terms of investigating outcomes later in the future. Next. So I really appreciate everyone's attention. I hope that you have taken an interest in DILIsym and RENAsym and our other products, and we'd love to take any questions that you have at this time.
Thank you, Brett. Great job. A reminder, if there are any questions, you may send your written questions using the question pane, or you may use the hand-raising icon on your control panel to ask your question directly. Please be sure to enter in your unique audio PIN. So going to the questions now, we have, is it possible to simulate 3 compounds simultaneously within DILIsym for DILI-DDI predictions?
Yes, absolutely. You can simulate 3 parent compounds. You can also simulate a combination of metabolites and parents, and actually you can simulate more than 3.
Okay, great. All right, so it looks like that's the end of our questions for Brett. Thank you again. We'll see you at the bottom of the presentations with the posters. So next, we have another quick question for you. Are you currently using QSP modeling technology to assist in your research activities? The polls are open now, and again, we'll give you 30 seconds to answer. All right, a little less than 5 seconds to go. The poll is now closed, and it looks like 62% of you are in the right place to continue learning more about our QSP modeling technology with DILIsym. Now, the first poster presentation today is by Vinal Lakhani, postdoctoral fellow at DILIsym Services. And Dr. Lakhani's role is to improve SymPup's capability, including size and tools across all QSP software.
Hello, everyone. My name is Vinal Lakhani. I'm a scientist 1 at DILIsym Services. I am part of the project teams that work on developing the DILIsym software, the RENAsym software, and the RENAsym software, as well as client-based projects. And those client-based projects is where we're going to start today. This project was brought to us by Pfizer some years ago. It's for a compound that they had in development for the treatment of epilepsy. Unfortunately, it was terminated after failing phase 1 clinical trials because it did show some volunteers with elevated ALT and thus led to liver safety concerns. So that's some of the background. An additional point of background is this compound was studied preclinically in rat species, and in rats it actually showed no elevation in ALT, and so it seemed safe from a preclinical standpoint. But then when they went to humans, it did show that elevation, so had to be pulled. The paper discussing the human clinical results are presented here in this Aleo 2019 paper, which I have a reference to at the end of my short presentation here. From our side, from DILIsym side, we would like to investigate why there was a differential response between rat and human, and what mechanisms are at play with this compound that perhaps led to those differences in species difference response, I should say. in the methods, for DILIsym's project, you tend to, the software requires a few pieces of input. One input is in vitro data, which investigates, which reports how toxic the compound is with respect to 2 or 3 different mechanisms of common mechanisms of liver injury. So 2 of them are shown here. On the left-hand side of this figure, I'm showing BSEP inhibition assay. And so on the top is the rat BSEP response, and then the bottom is the human BSEP response. So you can see in both species, this compound does seem to inhibit BSEP to some degree at a fairly high concentration. And on the right-hand side, I'm showing seahorse data, seahorse results, which investigates mitochondrial dysfunction, how much this compound actually injures mitochondria. And so this compound was actually investigated. A couple other assays were done with respect to other bile acid transporters, NTCP, MRP3 and 4, as well as the rat analogs. But I'm only showing the ones that had any kind of signal here with respect to those toxic assays. So we did not see any response for NTCP or MRP3 or 4 in either species. So just presenting the results for BSEP and the seahorse. So that's one set of information to know how much toxicity the compound is actually causing. And the second piece of information is how much of the compound is actually getting into the liver. And so on the right-hand side, I'm showing results from our PBPK simulation, which shows how much of this, which helps determine how much of the compound gets in the liver. On panels A and B, I'm showing the rat results. This is rat single dose simulation, and this is rat multiple dose simulation. This is the results after 28 days of daily dosing in rat. Panel C and D show the results or simulations in human. And the data points here are from preclinical species results, and then here is the dots here also show from the clinical results. Again, the top here is for single dose response, and the bottom for humans here, we did a simulation of 14 days BID, and this is the last day, or the last dose. And then panels E and F just show the liver concentrations, which can't be measured clinically, but this is just the simulation results predicting how much exposure there might be in the liver. So based on these simulation results, we can see that our PPK model does a really good job of capturing the observed exposure in both liver and plasma. Okay, so taking those 2 pieces of information, the in vitro toxic results and the PPK model, we put that together in the DILIsym software and simulate both rats and humans. So you see in rats that they're in the eDish plot here showing peak ALT in rat versus peak bilirubin. There's very few rats, if at all, that have any kind of response in either axis. And so recaptures the preclinical results that the compound appeared safe in rats. Whereas in humans, our simulations show that there's many humans here that have simulated humans that had peak ALT elevations above 3x. And in the worst case, some humans that were simulated also had elevations in bilirubin, which indicates Hy's law, indicates that they're in the Hy's law region, which is very dangerous. Table 1 shows a little bit of a further breakdown, comparing our clinical results in the top line to the our simulated results in the top line to the clinical results in the second line. With DILIsym, we can actually dig a little further. We can investigate, how much sort of bile acid accumulation there is in the liver. So, for example, for A and B here, or in the rat simulation, panel A shows the amount of bile acid accumulation in the rat liver, and you can see there's a range of response because of the different simulated rats that we had. None of them have any major deviation from their baseline once throughout the duration of the simulation. And then panel B here shows the ATP levels, and again, although there's a range in response by individual rat simulated, there's no major deviation from their typical baseline. Whereas in humans, panel C and D here, you can see that there's pretty high elevation in bile acid accumulation in human liver. And some individuals actually, simulated individuals actually take off, and these eventually terminate as simulated deaths. And it's reflected here in panel D, which is the ATP levels in the simulated humans. And in those humans, again, they plummet to 0, indicating a simulated death here. One thing to note is at time 14, there's a sharp change in the dynamics because that's where we ended our dosing. So we had 14 days of BID dosing and then 14 days of no dosing, just a follow-up simulation to assess the recovery. And although we didn't do any major quantification here, at least from visual inspection, you can see a couple of them do recover slightly here. So that's other information that we can glean using DILIsym software. Another piece of information, which is not shown here, but it is in the poster that will be available, is that we can investigate each individual mechanism of injury. So we have the bile acid. This compound seems to cause bile acid inhibition, and it has a response in mitochondrial dysfunction. So with DILIsym, the software, we can actually turn each. So these simulations here have both mechanisms on as expected in real life, but with the software, we can actually pick one mechanism to turn on and one to turn off to investigate which of these mechanisms is actually responsible for the injury. And so what we actually found was that if you turn the bile acid off and just have the mitochondrial dysfunction on, then there's no toxicity. Similarly, if you just have the bile acid accumulation mechanism on, but no mitochondrial dysfunction on, then you still see no toxicity. So it's very interesting that you need both mechanisms, both the bile acid inhibition and the mitochondrial dysfunction, to produce the observed hepatotoxicity in humans. So it's another one of the powerful strengths of our software. Another additional point here that I have is that the BSEP inhibition that we saw on the previous slide, the KI was around, the IC50 was around 311 micromolar. And just that data point alone, typically you don't think that this compound is going to produce any major bile acid related hepatotoxicity. It's a pretty high IC50, and so it might not trigger that mechanism. However, with our simulations with DILIsym, DILIsym shows us that while it by itself is not enough, once you add that extra mechanism of mitochondrial dysfunction, this compound does in fact lead to hepatotoxicity in humans. We did further investigation. Again, we're really interested in knowing why there's a difference between rat and human a differential response. And we found that the exposure level in humans is a lot higher. The compound is much higher exposed in human liver than rat liver. And again, because this is a software, we can artificially increase the amount of compound exposure in rat to see if we get any kind of response. And so when we did those simulations, we did see some mild aggravation, some mild elevations of ALT in rats due to the mitochondrial toxicity or mitochondrial dysfunction. Yeah. And so in conclusion, some of the major strengths of DILIsym software is shown with this project. We can compare across species. We can show that although this compound seemed clean in rats and seemed safe based on preclinical information, it actually, our software at the same time predicts that this compound is going to be toxic in humans. And so, hindsight's 2020, but hopefully this can be used in future projects that had this compound come to us before going to clinical trials, we might have given some evidence towards, given some pause to the project team to understand, to do some further investigations in humans before taking it to clinical trials. So, it's another reason to come to DILIsym services at any stage of your project. The second reference here is this work has been published with this reference here. So thank you again for watching my presentation. You can find my poster and others like it in our resource center at www.simulations-plus.com. I look forward to taking any questions from you.
Great presentation, Vinal. All right, so let's go straight to the questions. I see hands up. All right, so here we go. The DILIsym simulations predicted a non-trivial number of responders to this compound. Were you able to gain any insight with respect to any common physiological characteristics of this group of responders?
Yeah, so we looked at the group of individuals that responded and did some basic linear regression analysis and found that those simulated individuals, their parameters that were sensitive were actually low body weight, and so the individuals with very low body mass are the more sensitive ones. And then also related to the mechanism here, individuals that had some parameters that showed, that indicate the capacity for bile acid transportation. If those parameters were decreased, so individuals that had lower capacity for bile acid transport, they were also more sensitive. And that pairs with the mechanism here. One of the mechanisms is that it pairs with one of the mechanisms of bile acid transport inhibition. So yeah, the information is further, that's a great point. It can be found in the paper as well. So I encourage everyone to take a look at that paper. There's a lot of other cool results there too.
All right, can this be used with different drug batches?
I guess I'm not entirely sure about drug batches, what is referred to by that.
Okay.
Sorry, yeah, if there's a clarification later on, we can come back to that.
If the drug batches were produced by a different company and to demonstrate batch effect?
I see. Yeah, I mean, I think we typically take, the information from the in vitro studies. So let's see. Yeah, if you have different in vitro results from different batches, for example, then absolutely we can do differential simulations there. So if you have first batch produced and you put that through your in vitro assays and get one set of results and a different batch produces a different set of in vitro results, then yes, absolutely we can take those two different parameter sets and run different simulations to see how different or how sensitive batches are, different batches are. Yeah, absolutely. That's a very clever question, actually.
Great question, Shuchin Ma. If you need further clarification, you can always email us at info@simulations-plus.com. Thank you, Vinal. We'll now move to the next presentation by Yeshi Gebremichael, Scientist 2 at DILIsym Services. Dr. Gebremichael's research has been focused on the evaluation of DILI risk for several compounds with the DILIsym modeling software, in addition to developing the new modeling software, RENAsym.
Okay, hi. My name is Yeshi Gebremichael. I'm Scientist 2 at DILIsym Services Division. Primarily, I work on RENAsym software development project, but I also support other projects related to liver toxicity and heart failure. Today, I'll be presenting our effort on quantitative systems toxicology modeling of cisplatin-induced nephrotoxicity. So in this effort, we used in vitro assay to guide the mechanism of model development, and so I'll be presenting our effort in this direction. Okay, so to give a little bit background, cisplatin is one of the drugs that cause nephrotoxicity, which in turn leads to what's known as acute kidney injury through various cellular mechanisms like mitochondrial dysfunction, oxidative stress, and other mechanisms. So the acute kidney injury mechanism, either from cisplatin or many other compounds, is not fully understood yet. So quantitative system toxicology modeling is a tool that allows us to understand the mechanism behind acute kidney injury through mechanistic representation of the different toxicity pathways. So in this brief presentation, I'll briefly show you our ongoing effort on QST modeling development, and especially we're using cisplatin as our exemplar compound, and also we collected in vitro data to build the model. So I'll be showing you in the next couple of minutes, our effort in this area. Okay, so what you see here is an overview of RENAsym mechanistic model. So on the top, the right figure, you see the overview of the mechanistic model. And this model starts with drug exposure. So we have, a portion that describes the kinetics of the drug exposure. And we use GastroPlus for drug distribution. And now when the drug gets into the body, especially in the proximal tubule cells within the kidney, it causes, it affects different, it triggers different mechanisms. And some of those mechanisms are described in here with arrow. For example, you can see it increases the oxidative stress in terms of reactive oxygen species, or it affects, it causes mitochondrial dysfunction, or the drug may cause mitochondrial damage, DNA damage, or it can also cause, crystal nephropathy, which may affect distal tubule and so on. But if you lean on the right side of the flow diagram, what you see is all the reactive oxygen species or mitochondrial dysfunction. They lead to cellular death. In this particular case, is proximal tubule cell death, and that may trigger immune responses, and that by itself may cause further death on the cells. And the ultimate effect of that is there are some biomarkers that are released in the urine or in the blood, which we're going to be tracking, but also it may affect the downstream effect that that's related to tubuloglomerular responses. And all this is intertwined, and we're developing a model that describes these different pathways and be able to connect drug exposure with those downstream pathways and effectors. And we're measuring the effect of the drug on the proximal tubule cells through biomarkers that indicate for us the damage of the drug. So in order to build the model, the first thing that we need is we need to parameterize some of the mechanisms that are described in the in the arrow here. So two of the mechanisms that we are currently working on is the oxidative stress, the one that is showing in the arrow as reactive oxygen species, and also the mitochondrial dysfunction. In order to be able to describe these two key mechanisms, we need to have the data that's unique to the drug. For example, when you have cisplatin causes oxidative stress differently from another drug. And in order to be able to quantify the effect of cisplatin or any other drug on oxidative stress or mitochondrial dysfunction, then we need to have some in vitro data that would help us guide, parameterize that the effect of that drug. So we collected in vitro data that was measured by Nephrotoxicity and using renal proximal tubule epithelial cells, cisplatin incubated with proximal tubule cells, and then we measured some responses, for example, oxidative stress using high content imaging or oxygen consumption rate using Seahorse XF analyzer. And the graphs that you see on the left, the two figures correspond to the measurement that are done either using high content imaging or Seahorse. So on the very left, the oxidative stress parameterization is done using data that came from high content imaging. So the black circles are the actual data, and we fitted this data using our model, and the red line with the circle is the simulation result that is fitting the data in order to extract parameters that describes the oxidative stress arising from cisplatin. And on the right is the seahorse data, which is oxygen consumption rate. And this data shows that there is electron transport chain inhibition. And you can see the black one is, again, the data, the measured data. And the red is our fitting using our model. So once we fit the data, then we extract the parameters. And these parameters are shown on the right hand side, and that is fed into DILIsym. And then I can quickly show you the results of this. Then we run simulation, and in this slide you're seeing the results of the simulation and also the intermediate processes that are involved in our model. So you can imagine there is a compound exposure that causes oxidative stress, RAS increase, or mitochondrial dysfunction, which leads to ATP decline, or triggers caspase that leads to cellular death and eventually leads to biomarker responses. And what you're seeing in the plots are these different pieces of the puzzle being measured. So on the very left you have compound exposure, the proximal tubule cell concentration profile, and that leads to oxidative stress and mitochondrial dysfunction, and that eventually leads to ATP decline and cellular death, and eventually the biomarker release. So you can see, the drug releases a biomarker, and we get a signal that the drug causes toxicity. So in conclusion, our model was able to predict cisplatin-induced toxicity, and we were able to quantify alpha GST elevation. Alpha GST is a key biomarker that can tell us there is a cellular death, and the one that we're measuring is the urinary alpha GST, the urinary biomarker that indicates cellular death. And we did dose exposure analysis, and as you saw in the previous slide, under 533 milligram per meter square IV infusion, cisplatin led to 14-fold change in alpha GST. And when we are running under 100 milligram per meter square IV infusion again, then we saw 7-fold increase in alpha GST. And the 100 milligram per meter square study is in qualitative agreement with a clinical observation, which was found to be about 3.4-fold change. In our simulation, we found about 7-fold increase. So we're still developing the model, and there are additional improvements that are being currently done. But in its current states, it definitely shows that RENAsym shows it's a promise in combining QST modeling and in vitro data in order to be able to predict drug-induced acute kidney injury prediction. So the model is doing reasonably well, and that's the data we have. And so thank you for listening, and if you have any question, I'll be happy to answer. Or you can reach us, at www.
Okay, Dr. Yeshi Gebremichael was unable to be with us today, so Nadir Hamzavi will address today's questions. Dr. Hamzavi's research has been focused on developing new modeling software, RENAsym, to develop multi-scale quantitative system pharmacology, toxicology modeling of drug-induced acute kidney injury. And the first question we have for Dr. Hamzavi is, why is urinary alpha GST considered as good a biomarker for renal toxicity?
Thanks for the question. I'm happy to take it on behalf of Yeshi Gebremichael. So alpha GST is a constitutive protein that is released into urine in the presence of site-specific renal tubular injury. So when proximal tubular cells are injured and the damage lead to necrosis, they release their intercellular alpha GST into urine. And it's a good indicator of cellular death, specifically necrosis of proximal tubule, which is the primary target of renal injury. And we use this biomarker for modeling validation.
Great. The next question is, does the cisplatin PK or PD simulation account for all methyltheliin levels? Copper levels are also dramatically affected.
I think I'll think that I don't have a quick answer to the question at the moment. I will be passing the question to Yeshi Gebremichael, who can talk about the PBPK modeling in depth.
Okay, Rob, we'll get back to you with an answer for your great question. Thank you. And now going into the final poster presentation done by Brett Howell. Again, we're going to follow up Brett's presentation with the DSX demo, so stay tuned. Here we go, Brett.
Hi, my name is Brett Howell, and I'm the president of DILIsym Services Incorporated, a Simulations Plus company. And today, I'd like to present to you our work around the modeling of acetaminophen as it relates to its potential as a carcinogenic risk. So this poster is entitled Quantitative Systems Toxicology Modeling of the Acetaminophen Cellular Pathways for Liver Toxicity, which supports that acetaminophen is not a carcinogenic hazard in humans. So this work actually originated out of a request for the California Office of Environmental Health Hazard Assessment, commonly referred to as OEHA, to assess and review acetaminophen as a potentially carcinogenic risk to the public. Now, as many of you may know, acetaminophen has a long history of safe use at therapeutic doses. But it is well known that acetaminophen can cause liver injury at very high doses. And in fact, acetaminophen is one of the most highly cited causes of acute liver failure in the United States and in the world. However, almost all of these acute liver failures are occurring in cases where acetaminophen is being taken at doses that are much higher than the recommended dose levels. This quantitative systems toxicology work supported this review as part of a broader scientific weight of evidence assessment of the carcinogenicity hazard potential of acetaminophen. There were actually several different work streams that were conducted as part of this process, and those will not be presented on here, but will be published alongside this work in the future as part of a collective article or set of articles. So our approach was to start with DILIsym, which is a validated model of acetaminophen exposure, metabolism, and cell death or liver injury. DILIsym, which is shown schematically on the right-hand side of the slide, is actually a mechanism-based model that puts together a drug's exposure, metabolism, and distribution with the interaction of the drug and its metabolites with several pathways within the liver that can lead to toxicity. Those include such things such as reactive oxygen species, mitochondrial dysfunction and toxicity, and also intracellular bile acid transport and homeostasis. For acetaminophen, the oxidative stress or reactive oxygen species mechanism is most important. Now, DILIsym has been used many times over the last 10 years to address potential drug-induced liver injury concerns for novel molecules in drug development. However, originally, DILIsym was built upon a model of acetaminophen as a part of a CRADA with the FDA. And so for this reason, DILIsym is very uniquely suited to address this question of the mechanism of action of liver toxicity for acetaminophen and how it may or may not lead to carcinogenicity. Now, it was very important that we qualified and validated the model for use with acetaminophen before conducting this analysis. So you can see on the bottom right a table which is present in the poster, which shows the validation endpoints for different areas of the model, including the ADME and PK, glutathione depletion, hepatotoxicity, oxidative stress, mitochondrial dysfunction and inhibition, and also protein adduct formation. And what the table shows is that we have actually identified data supporting both the kinetics and the dose response aspects of the model. And we've cited those in the publications listed on the slide. And as an example of that, if you look to the left, you will see time course data in mice where acetaminophen was given, and two things were measured concurrently. First of all, liver glutathione, which has been depleted over time, is going down as you move to the right. In other words, as the acetaminophen is ingested and processed by the mice. At the same time, the liver NAPQI or reactive metabolite products of acetaminophen are beginning to go up. So we can see the sort of behavior where the buffer for the liver is depleted and at the same time harmful products are produced. And what you can see in the small inlay is that the DILIsym model reproduces this behavior. We have an increase in the NAPQI protein adducts and we have a decrease in the liver GSH. So this is just one example of how DILIsym qualitatively and quantitatively reproduces many of the data sets that are in the literature for acetaminophen. So with this in mind, we conducted a series of simulations to help specifically address the question or the concern of carcinogenicity. Through that analysis, we came to several conclusions. First of all, clinically relevant exposures of acetaminophen do not lead to meaningful cellular stress that would cause carcinogenicity. Second, at higher doses, when cellular stress does occur, the buffering capacity is depleted and cell death also occurs. Lastly, simulations including population variability also support that across a wide array of patient backgrounds, acetaminophen exposure only results in significant stress or oxidative stress in the cell or DNA effects under conditions that also cause cell death. Now, to illustrate that, we can look at the two figures shown on this slide. First, we have at the top an example of acetaminophen dosed at 4 grams per day, which is a therapeutic dosing level in a population of individuals. And we can see that even across a wide range of individuals, the glutathione levels stay reasonably high, and there are no depletions of glutathione at that dose level. What that means is that we have a very low amount of mitochondrial stress, illustrated by the red lines, and very minimal to no liver cell death, illustrated by the lack of drop in the orange lines. Now, if we go below that, we can increase that dose just by 3x, so 12 grams per day. And now we see a series of concurrent events. We see some of these simulated patients having glutathione be depleted. At the same time, as their glutathione is depleted, we see mitochondrial impairment increasing. And then subsequently, shortly thereafter, we see the liver cell death increasing or the viability going down, as indicated in orange. So we can see these concurrent events in time kinetically, and this shows us that really with acetaminophen, you're either in one of two zones. You're either in the safe zone where there's minimal stress, or you've got so much stress that you get liver injury. And we can also see this as a function of dose to the right. So here we have not time, but dose on the x-axis. And in this case, the simulations are conducted more in line with the clinically relevant scenario of a single dose of acetaminophen. Certainly in the case of overdose, patients tend to take a whole bottle of acetaminophen at one time. And so what we notice is that if we're down on the therapeutic side of acetaminophen, there's very minimal cell stress. You've got glutathione, which is represented by the green region of the figure, if you will, across a whole population, where almost all of the simulated patients have substantial levels of glutathione remaining. They have minimal mitochondrial stress, as shown with the red shaded region, and they have minimal cell loss. But as you move to the right, you see glutathione beginning to trend down. Certainly by 10 to 15 grams of acetaminophen, you're in a zone of significant depletion of glutathione. And so you start to see mitochondrial impairment increase. And very shortly thereafter, starting roughly at around 15 grams, but certainly by 20 and 25 grams, in that range of 15 to 25 grams, you begin to see the minimum viable hepatocyte fraction go down, or basically cells dying in the liver. And so what this shows us is that there's this transition that occurs as you're increasing the dose. And actually, the transition occurs in a reasonably narrow window. You don't have a large number or a large margin, if you will, between safe acetaminophen use and liver toxicity. And that's really important because the concern for carcinogenicity is that there's a region where patients are subjected to stress, but their liver cells are not dying, thus the stress potentially causing carcinogenicity. What we see here, though, is no evidence for that. In fact, we see a nice sharp transition from safe to unsafe for the cells and ultimately for cell viability, which avoids the situation of a continued chronic stress. Now, what's nice is you can see with the, certainly with the black, but also I'll draw your attention to the white circles, which is published data showing bilirubin rises, which are very indicative of liver function, liver dysfunction, liver failure, going up around the same time, 20 to 25 grams in terms of the percent of patients reported in the study who have increases relative to our simulation. So again, another key piece of validation showing that the modeling results are validated and relevant compared to clinical outcomes. So to summarize, simulation results and mechanistic experimental data support that at therapeutic acetaminophen doses, cellular glutathione deactivates the reactive metabolite, also known as the NAPQI metabolite pathway. And there is sufficient buffering capacity to prevent any meaningful protein adduct formation or oxidative stress. However, following overdose of acetaminophen, cell death occurs before any adverse conditions occur. So for example, before you could have something like DNA damage or strand breaks that could ultimately result in carcinogenicity. So ironically, as we conducted this analysis, we came to the conclusion that acetaminophen is almost designed, if you will, or serendipitously designed to be safe from a carcinogenic risk because of the fact that there's such a sharp transition between safe use and liver failure or cell death in the liver. These results collectively support that acetaminophen is not a carcinogenicity hazard to human health under any conditions, including the therapeutic doses or higher doses beyond that level. If you have any questions on the content of this poster, please email us at info@simulations-plus.com with questions. Thank you for watching. You can find my poster and others like it in our resource center at simulations-plus.com. I look forward to answering any questions now.
Thanks, Brett. It looks like we already have the first jump on a question. Why does APAP transition so abruptly from a safe drug to a liver toxicant as the dose increases?
Thank you, Arlene. Can you hear me now?
We can hear you now.
Oh, I'm sorry about that. Yeah, so the answer to the question is simply related to glutathione. It's the fact that glutathione is the buffer for acetaminophen, acetaminophen's reactive metabolite, and it's the rapid depletion of glutathione at higher doses that leads to that quick transition. So it's an interesting sort of chemistry, the way that glutathione acts as a sink for that reactive metabolite.
Okay. Christy asks, she says, great presentation. Is DILIsym also able to do modeling of antimalarial drugs such as hydrochloroquine?
Yes, absolutely. We can represent those types of drugs simply by inputting the correct in vitro data and doing the PBPK modeling for them and inputting that into the software. Yes.
Okay. Let's see. And another question, are you seeing increases in the number and quality of biomarkers, either directly in your research or with collaborators, that both help validate your DILIsym and RENAsym modeling results, as well as provide better clinical diagnosis?
Over a number of years, we have continued to see advancements in the biomarker space, and we do continue to monitor those advancements and input those biomarkers mechanistically into our software. So it's certainly something that has informed our modeling, helped validate our modeling, and that we will continue to stay focused on. It does move at relatively slow pace because you're talking about clinical studies and the release of clinical data, oftentimes from consortia or clinical trials, which take time to become unblinded and then published or released to the public. But yes, we've certainly have a large program internally underway in terms of representing and understanding biomarker responses.
Thanks, Brett. And now what's next for the DILIsym team? Well, Corey Berry, Senior Software Engineer, will take us through the highly anticipated DSX software release. Questions related to the demo can be emailed to info@simulations-plus.com.
Hello, welcome to the DILIsym X software demonstration. My name is Corey Berry, and I'm a Senior Software Engineer at the DILIsym Services Division of Simulations Plus. Today I'm going to give a quick demonstration of the DILIsym X software platform in its current version. It's important to note that this software is currently in development and some aspects of it may change in the final release. So when you first launch the software, you're presented with a license screen that shows you your current license status. Here you have a license status of active or inactive, and also how many days you have left on the license. You also can be presented with additional details related to your license. This is the DILIsym X main dashboard. From this window, you can access different links to online DILIsym resources. Here on the right-hand side, there is a RSS feed that's connected to the Simulations Plus website. And so you can get latest updates from the Simulations Plus website right here in the application. So currently, what I'm going to show in this demonstration is a simple SimSingle. Right now, there are no projects loaded. This version of DILIsym operates on the idea of projects. Each project contains multiple configurations. The configurations are located here on the left-hand side in this toolbar. So the first thing I want to do is just create a new project. I'm going to call this one Sample Project. You'll notice at the top that the title bar changed from it changed to DILIsym X Sample Project. That lets you know that a new project has been loaded. And you also have the ability to put notes within an individual project, as you can see here at the bottom. So now the next thing I want to do is create a new configuration that'll be stored within this project. So I'm going to click SimSingle. And right now there are no configurations available, so I'm going to click the Create New Configuration button. I'm going to call this one Sample SimSingle. Now, the new configuration is loaded and stored within the project here at the top. As you can see right here in the middle, there are several buttons that allow you to change which parameter set is loaded within the SimSingle. Right now, we just have the human blank parameter set loaded for many of these parameters. So you have the species, calorie, drug, solver, and the 3 different compounds, the time, and also ability to set the output panel and the specified data features in here as well. So just to demonstrate what happens when you click one of these, if I was to click the drug parameter, this gives me a list of different parameter sets that are stored within the program that I can access and apply for this actual parameter set. And if I click compound W, I get a different list of items that are selectable. So this allows you to mix and match different parameter sets to create your own personal sim single. For this demonstration, I'm just going to leave everything as it is. In order to run a sim single, there's this play button here on the left hand side that you can click. I'm going to go ahead and click that to start the dialog for running a sim single. This is a sim single run dialog, and as you can see on the left and right hand sides, I have different boxes that indicate the selections that you made in the previous window. Before the simulation runs, this kind of lets you double check that these are the things that you want to run. And so when you're ready and you're sure that these are the things you want to execute, you can hit the play button above. So I'm going to go ahead and hit that now. While the simulation runs, I'm going to explain what's here at the top. The system status window shows you how many threads are currently in your CPU. So on this computer, I have a 16-core, 32-thread processor, and that's indicated above. Since this is a sim single, I'm only going to use one thread. There's a progress bar showing how much RAM you're currently using. This is very helpful when you're running large simulations that could potentially produce a lot of data in your memory. It lets you know, if you're getting ready to run out of memory or not. On the right-hand side, there's the simulation status window, and it kind of lets you know the current state of the running simulation, how many sims have been completed, whether or not there are any exceptions or errors from the simulator. In this case, the simulation is finally finished. It finished in 5 seconds, and now I can go ahead and view the results. So if you click the View Results button, this will bring up a new window. By default, this is currently empty, but this will be changing in later versions of the software. This is the actual plot window that allows you to see the results from the simulation. So in order to initialize a blank plot for this, I'm going to go ahead and drag the time step variable over to the independent variable box. Once I do that, it's able to plot the data along the right here on the right hand side. So this first column that you see on the far left is a list of all the variables organized by group and subgroup. Or you can also just filter by name and select from all the variables available. Here in this middle column, we have a list of output panels, and each output panel has a list of variables already predefined, and that's what's getting plotted over here on the right hand side. So in this case, I'm going to change this to common outputs, which is one of the built-in output panels. And as you can see, it updated the plot. All right, I'm also going to show you that you can also select a new output panel, which essentially makes a blank set of variables for the dependent variables, and you can drag and drop variables from the left-hand side. So here I'm just going to drag these over, whatever variables you really want. You can multi-select as well and drag over multiple variables. So now I'm going to move over to the right-hand side and show you some of the features currently on the plot viewer. So you have the ability to change the background color, and you can select from a list of named colors, or you can create your own custom color. In this case, I'm going to select the dark color to let you see what it looks like if you completely invert the look and feel of the chart. And now I'm going to select a foreground color, and again, it brings you to the same list. I'm going to choose a light color this time. And now you can see it changed the colors for the chart. And there's also a feature to enable or disable grid lines, and to enable or disable the legend, as well as line symbols. Of course, this is a very minimum set of features for this chart. There will be plenty more in the final version that you can modify. Change this back. Okay. So now what I'm going to show you is that you can also change the font for the different labels on the plot. In this case, it's set to a default of 12 point, but I can change this to something smaller and even a completely different font if I wanted to. And as you can see, that changed the axis titles. All right, and so here at the bottom, you see there's a tab for chart and data. Currently we're on the chart view, but if I change this to data, you can see I could see the data behind the chart, and I'm able to scroll through it. This data can also be saved with this button here to an Excel document, or it could be, you can also save the chart to an image, or actually just copy the chart image as well. Over here in the middle panel, you can save a new panel. If you create a new output panel like I have here, you can save it as a new output panel and save it to your current project. You can clear the panel and also reset the variables located here. But as I said, we'll be adding more features to this window. This is just kind of a basic application to show the results from the SimSignal. All right, so I'm going to close out of this one and go back to the main application. All right, I'm going to move over to the settings. Currently, we have a settings window. And what this does is allow you to change performance settings, output settings, and even the theme and look and feel of the application. Here at the top is a CPU performance setting, and this allows you to set, based on a percentage, how many of your CPUs you want to use for simulations. In the event that you're running SimPops or multiple parallel simulations, you can assign how many you want to use, depending on whether or not you want to maximize the performance or just, minimize it. By default, it's set to normal, which utilizes half of what your CPU threads on your computer. There's also the ability to run individual simulations as processes or run them as threads. There are pros and cons to both of these approaches. In addition to the SQLite-based database output format, you can also save output results in Excel, JSON, tab separated and comma separated files. These will be additional files produced alongside the actual run configuration database file. And of course, you can change the default location that results are stored to. And finally, you can also change the look and feel of the application. Currently, this is set to this this reddish berry color to kind of match the DILIsym logo, but you can also change it to a multitude of different colors. And also, you can modify the look and feel of the toolbar as well. You can minimize the size of the buttons by removing the text and show only icons for the toolbar. You can hide the toolbar completely. And these settings persist throughout runs of the software, so it gets saved to a file. And if you also want to experiment with removing the title bar system, you can actually remove your title bar, so you can drag the window just by using the built-in title bar within the app. So that's another feature of the application. All right. So I believe that will conclude this current demonstration. And if there are any questions, please email them to info@simulationsplus.com. Thank you for your time, and we'll continue working on this product to improve it. Thank you very much. Bye-bye.
Great job, Corey. Thank you to our presenters and our participants today. Although some of our face-to-face events might have been canceled or postponed, we have many virtual programming opportunities that you can take advantage of. ranging from free webinars to virtual masterclasses to support your pharmaceutical research and drug development efforts. Join us on Thursday for day 2 of our live stream learning event, and you can find registration links and today's webinar replay at www.simulations-plus.com. Thank you for joining and have a wonderful day.